Thank you everyone for joining our JoVE webinar today. My name is Ron. I'm the Director of Editorial here at JoVE. And today we're joined by Rami Shahror from the University of Arkansas. And he's going to present today a stem cell-based therapy for traumatic brain injury. So, this is a very hot topic. It's been getting a lot of attention. In his original paper he published with us in 2019 has been getting a lot of attention. So we wanted to kind of host a webinar and answer any questions that people may have. So we will be recording the webinar and sending out the recording afterwards. So you'll have that for your records. And also we'll be having a Q&A at the end. So feel free to add your questions in the Q&A during the presentation, and we will address those towards the end. Okay. Take it away, Ronnie.
Thank you so much, Dr. Myers. It's my pleasure today to present part of my paper that I published in JoVE, and I would say it's a very nice experience for technology transfer and also technique transfer. So the data topic will be the title of Safe and Non-Invasive Method for Tagging, Delivery, and Tracking of Stem Cells into Brain of Traumatic Brain Injury Murine model. We know that mesenchymal stem cells are one of the potential stem cell therapy for CNS and other injury, and I will not speak in detail about the stem cell therapy, but we were interested in mesenchymal stem cells because they have a safer profile and it can be manipulated in vitro before transplantation. Also, the source can be isolated from different sources from a human. However, there is a limitation of using mesenchymal stem cells in a clinical setting, especially for brain injuries, that due to the poor survival and tracking of these mesenchymal stem cell at the injury sites, to the harsh microenvironment that occur at the injury sites in the brain. And other limitation is the brain injury complexity that needs more additional therapeutic potential to add to the mesenchymal stem cell to be more potent. Another special limitation for the CNS, I would say, is the blood-brain barrier. The stem cells generally need to bypass this blood-brain barrier, and that might limit the use of mesenchymal stem cell. Although there is some report that mesenchymal stem cell can bypass the blood-brain barrier, however, it's still considered as a limitation for therapeutic intervention generally, and mesenchymal stem cell therapy for brain injuries. So one of there is strategies that can be used to enhance the potency of mesenchymal cell therapy and facilitate use mesenchymal stem therapy in brain injuries by improve their homing and survival at the injury sites through the coating of the cells or genetic engineering of these cells to increase their homing ability to the injury site. And other strategy is using non-invasive delivery method and tracking methods for mesenchymal stem cell into the brain by, for instance, intranasal delivery of mesenchymal stem cell. It's a very novel method for delivery of stem cell generally, and it's a non-invasive method. So it could be applied in a clinical setting and could be repetitive therapy, stem cell therapy for the patients, since there is no surgical procedure involved. Another strategy is using SPO, Superparamagnetic iron oxides, nanoparticles, labeling, and that allow for tracking of the stem cell after the delivery. So we were interested in the last two strategies. Superparamagnetic iron oxide nanoparticle, or SPO, the behavior depends in many physical chemical properties of the SPO. Depends on their shape, size, and surface property that will affect their toxicity, the biodistribution, magnetic properties, and the internalization to the target cells. So always when we design experiment of labeling cell, generally using SPO1 and or stem cell specifically, we have to take in consideration these properties and take consideration these effects that may occur due to the labeling with SPO1. So the iron oxide or SPO1, the nanoparticle we have used, it's a commercially available called evergreen nanoparticles. And this nanoparticle have a core with a diameter of 8 nanometers and embedded, it has a dextran coating that embedded FITC. So that allow us to track this nanoparticle in the cell without doing any staining due to the FITC. And as we know that nanoparticle generally, especially the coated nanoparticle, they tend to aggregate to each other. And that might affect the labeling potency or internalization of nanoparticle into cells. So it's a good procedure or good steps always to do some the electron microscope scanning for this nanoparticle before do any labeling in the cells to make sure they have the right size, and also to confirm that there is no aggregation of these nanoparticles. Nanoparticles, it's reported that high concentration of nanoparticle could be toxic for the cells. So before establishing our protocol, we want to know what kind of concentration we could use for the cells without changing their behavior or changing their proliferation ability. Our cells that we have used are the mesenchymal stem cell isolated from bone marrow, and they're labeled with MCHER already, so we don't need any immune staining later on. And since the nanoparticle we have used already embedded with FITC, so we just incubated the nanoparticle at different concentration for 24 hours with these cells, and without any staining, we could see under the microscope, the first microscope, the staining ability and the internalization of this nanoparticle. So we could see there's dose-dependent internalization, I would say, of the nanoparticle into the cells, up to 25 microgram per ml for 24 hours. And that's the highest concentration we used, and it's based on the literature, so it's a safe concentration. So we use this concentration of the nanoparticle for further in our experiment, and to confirm that the FITC signal that we saw in the fluorescent microscopy is due to internalized nanoparticle, not aggregate nanoparticle. So nanoparticles sometimes aggregate into the surface of the cells, so it could be give false positive signal. So we perform confocal microscopy, and we clearly could see the nanoparticle inside the cells in the cytosol. Since this nanoparticle has an iron, that's advantage we can use for MRI imaging and later on tracking when we do that intranasal transportation. So to determine what number of the cells that we can detect, we're interested to see the lowest number we can use that still we can detect through the MRI. We use T2 star sequence. There's different sequence with different sensitivity. So we use the T2 star sequence, and we basically injected this labeled mesenchymal stem cell into gelatin, into a Bendreth tube. And at this Bendreth tube, we did MRI imaging using different cells number. So we can clearly here see in this image, like up to as low as 50,000 cells, we could see the hypo-intense area, which represent the iron, which represent, of course, the labeled mesenchymal stem cell, up to 150,000 cells in 1 ml of gelatin in the Bendreth tube. So we confirmed that we could detect this labeled cells in MRI. As we originally in our lab, we tested in TBI, traumatic brain injury, which is a damage to the brain resulting from external mechanical forces, or could be temporary or permanent physical or psychological impurities need to get that result from the TBI. So it's a hot topic, and TBI, it's a heterogeneous injury due to the different causes that result in TBI. It could be plus TBI, it could be TBI due to fall or car accidents or in the battlefield. So we were interested in TBI model generally, and the model we have used in our experiment, it's called controlled cortical impact injury model. Basically, we use the impactor, and this impactor will impact into the cortex of the brain after we do a craniotomy window, and you can easily control the speed of the impactor and also the depth of the impactor. Therefore, you can induce different injuries at different severities of the TBI. The injury we have used, we used 5 meters per second and 250 micrometer, I remember. And that's enough to induce TBI injury, and that results in tissue loss, as we can see here in the image. This is the experimental design for the paper we have published in JoVE. We basically labeled the mesenchymal stem cells with the SPO, the nanoparticles, for 24 hours at the 25 milligram per ml. And after the labeling of the mesenchymal cell, we washed them. Then we did intranasal delivery. For intranasal delivery into the TBI mouse, we used hydronase at 4 units per ml, and at different administration, for administration actually, with a total of 100 units per mouse. So we used the labeled stem cell into Neanderthal, the 2 of them, and we each time we do installation, we wait like 3 minutes up to 5 minutes, then we continue. Then we send this TBI mouse into the MRI scanning imaging. Since this mesenchymal stem cells, they already have the nanoparticle, we could see the nanoparticle as a green signal, since they have embedded with the FITC. And this FITC, as we can see in the nice confocal image, it will be inside the cytosol of the cells. Successful labeling of the stem cell, as you can see in D, the cell suspension will have a reddish color due to the presence of the iron inside the cells. So we perform MRI imaging at different time points, starting from 1 day up to 2 weeks. Now, since the cells have iron inside them, this signal will have like hypointense areas induced due to the iron presence. Some might argue the presence of the iron doesn't really indicate that the cell is still alive at the injury sites. At another study, we did sectioning, and we did immunostaining with the Ki-67, which is a marker for proliferative cells, and we could see colocalization between the FITC, which represent the iron and the nanoparticle, and Ki-67, which represent the proliferative cells, and the M-cherry, since our cells have already M-cherry. So we confirmed that these cells that we see now in the MRI images as hypointense areas, they are alive and able to proliferate in injury sites. Also, we take advantage from a software, it's called ATK Snap, this software used for segmentation of MRI images, originally used to study the morphology and the different part of the brain following the MRI imaging. So we took advantage of that as we have a different section of the MRI, and we could non-invasively at different time points for the same eyes, we could track the size of the hypotenuse area which represent the stem cells, and we could track their location. And we easily could detect the hypotenuse area at the injury sites, and we could also quantify the hypotenuse area and also quantify the injury region itself. So that indicate that there's successful homing of this mesenchymal stem cell after the intranasal delivery. It's important steps also to confirm the signal that have shown the MRI is not false positive signal, because some MRI sequences, they might have a background signal which would give us false positive signal. So one way to do it is using Prussian blue, since the nanoparticle has an iron, and the Prussian blue will label the iron, or will show the iron as a blue color in the tissue. This section at the injury site, and since already the cells have FITC in their nanoparticle, without doing any immune staining, we could detect also the FITC signal, and we could see it closer to the TAPI signal. So this represents the cells that able to be migrate through the intranasal delivery into the injury sites. So this confirmed that there's successful internalization of the nanoparticle inside the cells, and also successful homing of the labeled nanoparticle into the injury sites. Yes, the signal wasn't higher. It wasn't high as other method we have used before, but it's non-invasive method of stem cell delivery, and also you can easily manipulate the stem cell. Doesn't matter the source of the stem cells in vitro before the transplantation. And another advantage is you can administer different dosage of the stem cells at different time points for the same animal. And later on in a human, we believe that it could be very helpful, since you can do a stem cell delivery through intranasal route without a different time point or a different dosage for the same patient. So we're excited about the results, and we think that's a very promising method of stem cell delivery. But for a research purpose, it's always good to confirm the results you got, especially from the MRI, because it might get some background signals, and or you can get a false positive signal. So staining or sectioning after the delivery, it's a good way to confirm the results that you got. So thank you so much for your time, and please, if you have any questions, I would happy to answer them. Thank you. Yes, so I see here, is this technique also helpful to post-brain stroke? I think, yeah, TBI and stroke, they share many pathological events, including neurovascular injuries. And since we could detect the cells into the injury sites, yes, I think it could be applicable into the brain stroke, ischemic stroke or hemorrhagic stroke. I think both it could be applicable, since the TBI model we have used also can result into destruction of the blood-brain barrier, which is also presented in hemorrhagic stroke. So I think, yeah, that could be applicable. Another question, I had a right thalamus bleed and my left side is affected. I didn't understand what the question is. Could you repeat the question? That I had a right thalamus bleed and my left side is affected. I'm not sure what that. A question by Nazila. Thank you so much. Much appreciated information. I would like to ask, can they describe us the advantage of clinical usage of nanoparticle in comparison with the vectors in case of clinical efficacy? Yes, the nanoparticle that we have used, they are FDA approved. So that's one thing, say one advantage, very important advantage. And for using vector for labeling the stem cells, you still you need additional how to say IRBs before doing the clinical trial in a human. So using FDA approved nanoparticle, which is well characterized and well known in the industry or in the field, that make you avoid this using of vectors in stem cell therapy. A question by anonymous attendee: do you think the treatment will work for long COVID cognitive issues such as brain fog? I can't answer this question. I don't have the expertise about the COVID-19, but I think it works more with the brain injuries, such as stroke or TBI. A question by GC: I'm missing my left atrium due to TBI. Can stem cell regenerative tissue in right? There's a clinical trial using mesenchymal stem cells, and they published their results 3 years ago. And the result they got from mesenchymal stem cell therapy, although the route administration is different, they used a direct injection of the stem cell into TBI patient, and they had very promising results actually. I think there's also ongoing clinical trial for using mesenchymal stem cell for TBI, and there's more attention for using mesenchymal stem cell, as I said, due to their safe profile and feasibility for isolation from the same patient. It's possible to view the PET imaging instead of MRI. That's possible, but I think the MRI advantage is available everywhere, almost every hospital. And the MRI, you will have the advantage to use different sequencing. And depends on the sequence, you can manipulate the sensitivity of detection, the brain part, also the nanoparticle of the label cells. So the MRI, I think, is more visible than using PET for the current procedure. MSC can be used for TBI with hemorrhagic shock. Yeah, I answered this question. Yes, it can be used for hemorrhagic stroke. How train this cell functionality? Okay, for functionality of the cells in TBI after administration, as I said, it should be followed by some experiment to make sure these cells are alive. Sometimes you will be able to administer the stem cells into the injury sites, but they will not survive. And by that, they will leak their content, including the nanoparticle. So you will have a false positive signal. So it's a good confirmation study to have, I would say, immune staining for a marker for these stem cells and their proliferative efficiency. When they are alive, would be proliferate, and would be able to functional, would be able to differentiate another cell type. That's a very interesting aspect to do or to have in any stem cell therapy generally. And this treatment commercial availability, yes, can. now the treatment is not commercially available. The very commercially available is the nanoparticle that we use for the stem cells. I'm not sure if there's any commercial available center for using a stem cell. I can't recommend that or deny that, but I think there's some clinical institute or centers that they have or they facilitate stem cell therapy for different patient, different injuries. Thank you. Response, which is the idea investigation, some cytokine after before nutrition. Yes, I think investigating the cytokines before or after the administration of the stem cells, characterization of the stem cells due to the labeling is very important to make sure that they will not change their behavior, they will not change their cytokine profile, and also after administration, it's a good idea also to check their differentiation ability. Some might differentiate into other cell type. Some reported that mesenchymal stem cell will be able to differentiate to Murine at very small percentage. So that because also be detected after the administration. I think immune staining is one way to do it. How much is the approach effective against certain infection like fungal meningitis? I can't say anything about the microbiology. I don't have the expertise to answer this question. I'm sorry about that. But I mean, you can check the literature, and if the fungal meningitis has a neurovascular injuries, I think that could be applicable.
All right, so I think that's the last question. So thank you, Rami, for a great, fantastic presentation here, and thank you everyone.
Thank you so much
for all your questions and for attending. Like I said, we will send out a recording of this later in the week for your records. And yeah, well, thanks everyone. Thank you, Rami, and enjoy the rest of your day.
Thank you so much. Thank you.
Take care.
Thank you.